So, hello everyone, welcome back again to the Mycetoma Research Club. In today's session, we will be having Dr. Lamis Al-Khair. She will be talking to us about reproducibility and she will take us through the world of reproducibility. Lamis is a pharmacist. She's a lecturer at the Faculty of Pharmacy at the University of Khartoum. She's also a researcher at the Mycetoma Research Center. And she's an advocate for open science. She works as the Director of Training and Development at the African Reproducibility Network, AREN. So Lamis, the floor is yours.
Thank you so much, Samira, and it's always my pleasure to present in the club, and I hope you will enjoy today's session. So let me share my screen. Okay. Can you see my full screen?
Yes, we can see it, and it's always a pleasure to have you.
Thank you so much. So hello everyone, good evening, good afternoon, good morning, depending on where you are at the world. And as Samira said, I'm Lamis Al-Khair. And I'm a lecturer at the Faculty of Pharmacy, University of Khartoum, and I'm a researcher at the Mycetoma Research Center. I'm also Director of Training and Research Development at the African Reproducibility Network. And I'm really happy to be with you today to talk about one of the principles of open science, which is reproducibility. So in today's session, we'll go through a journey about what is reproducibility, what is causing issues with reproducibility, and how we could possibly solve it. So before I start, I wanted to give a brief overview of AREN. AREN is the Africa Reproducibility Network. We have a relatively small team with Emmanuel Bache being the founder and executive director. I think he's here in the audience with us today. So thank you, Emmanuel, for joining, and me, and then we have regional coordinators for African regions. So just a brief overview of AREN. AREN is a community-led initiative. It is founded and run by African researchers, and its main aim is to advocate for open science adoption within African institutions and African communities. As I said, our mission is to empower African researchers and institutions with the knowledge and skills and resources needed to meet the global demand of openness and reproducibility in research. Yet, we're actually even catering to communities outside Africa through this initiative, the Open Bridges Initiative, where we also go and engage with communities outside Africa. So even if you're outside Africa, you're still welcome to join our community. So our agenda is basically to promote open science and reproducible research in Africa. We are mainly targeting grassroots researchers trying to build their capacity, and we're supporting the establishment of local reproducibility network and hoping to set a continent-wide open science community. Just a brief overview of our community. We have community members from over 30 African countries. I think we even now have more than 600 members in our community with more than 200 institutions. They come from so many different disciplines. They have many different positions or roles. And we value and embrace each and every member of our community because we think that each member is a gateway to their local research communities through which we can be able to reach those local communities and inform them about open science knowledge and practices. We have a variety of open science engagement activities. We have community trainings. We have the local network leads training program. Actually, Samira was part of the first iteration of the program in 2024, and she's now one of our local network leads. And we also, in this iteration, the second iteration was just started April 2026, and we have Lubna also from the MRC currently joining this cohort. So it's exciting to see that we're going to have more local network leads within the Mycetoma Research Center. The Then we also have the RN Echoes webinar series, where we invite African researchers to talk about their experience with open science. We have the Open Science Bytes resources, which are open educational resources that you can find on Zenodo, that you can download and use and reuse openly. We're also trying to work on national and discipline open science working groups. And we also have the Open Science Bridges initiative. And this is this presentation is part of this initiative, where we go to communities and interact with them and teach them about open science. So the main idea is that we want to move open science from just being abstract to being more practical and relatable, and hence we have all of these open science engagement activities. So this was just a brief overview of AREN and our activities, and of course everyone is welcome to join. Samira, if you could add the link to the sign-up form. We have a simple sign-up form that you can fill in, and then you'll be part of the AREN community.
I sent it in the chat. I sent the sign-up form. So feel free to sign up and join the community.
Thank you. Thank you so much, Samira. So moving to today's presentation agenda, we'll have a brief introduction about reproducibility. We'll define some key terms. We will try to understand the reproducibility crisis and see the history of it. And then we'll see open science as a solution. We'll also discuss some of the current efforts and progress that is happening, and then we will end up with some key takeaways. So what is reproducibility? So reproducibility is the ability for a study or an experiment to be repeated by other researchers using the same methods and data and tools used by the original study to obtain the same results. So it's basically a group of researchers would take a study produced by another group of researchers. They use their same data, their same methods, their same tools, and they should come up with the same results. If they come up with the same results, then we can say that this study is reproducible, meaning that this results did not come by chance, for example. So these results are really robust. And reproducibility is actually one of the most important principles of open science. Almost all of the open science practices are revolving around reproducibility, or their ultimate goal is that research is reproducible. In many cases, you'll see that the term reproducibility and replicability are used interchangeably in open science discussions, yet there is a difference between them. So I thought it might be important to differentiate between them. So we have both reproducibility and replicability are aiming at obtaining similar conclusions or findings. But in reproducibility, we use the same data and the same analysis to test the consistency of the findings using the original setup. Like, are the findings consistent? If we use the same data and the same analysis, do we come up with the same results or not? While in replicability, we can use different data and do the same analysis and then see if the results are robust. For example, if a study was done in a community in Europe, then maybe we want to replicate the study in a community in Africa. And then we see if we obtain the same finding, this means that these findings are robust across different conditions. Reproducibility is mainly aiming to ensure the accuracy and reliability of reported findings, and replicability is mainly testing the generalizability and validity of the finding across different conditions. So it's important to differentiate between these two, but nevertheless, in open science, you would hear these terms be used interchangeably. So this is a nice, for the visual learners, it's a nice infographic by the Tuning Way. As you can see here, we have reproducibility. We have two scientists that are using the same data and the same analysis with the aim of reaching the same conclusion. While in replicability, We have different scientists that are using different data, yet they're applying the same analysis. And again, they're also hoping to get the same results so that we can say that the results are generalizable. So the question is, why is reproducibility important? Why all the fuss about reproducibility? Reproducibility is important because it upholds trust and credibility in scientific findings. That's the main issue. If results are reproducible, then the scientific findings, the results can be trusted and are credible. And it's also important because as scientists, we build on the results of other scientists. So if we have reproducible results, then this means that we have a dependable foundation for future research. And this forms different importance for different stakeholders. So we have the scientific community, and this is important for the scientific reproducibility is important for the scientific community so that they can easily build on results. For the public, reproducibility is important because it's needed for them to avoid misinformation. For the funders, reproducibility is important because it gives them informed data on where the resources should be allocated. And for policymaker, it will help them in making informed decisions related to public policies, national policies, or even international policies. So reproducibility is really important, and it has different importance depending on the stakeholders that we're looking at. We cannot talk about reproducibility and replicability without talking about rigor. And rigor is a central part of reproducibility and replicability. And when we say rigor, we refer to the strictness, precision, and careful execution of research method. If a method is rigor, it means that it's strict, it's precise, and it has been executed carefully. What rigor does is that it ensures the validity and reliability and accuracy of scientific investigations. And a rigorous study would follow a well-defined methodology. It will use appropriate statistical techniques, and it will avoid biases. So these are the general characteristics of a rigorous study. And again, as we said, rigor is, if we have rigor, then we will have reproducibility and we will have replicability of scientific findings if we follow rigorous methodology. So you might be wondering, where did this all start? And this started with the reproducibility crisis. And the reproducibility crisis refers to significant challenges within the scientific community where it is becoming more and more difficult or even impossible to replicate or reproduce many scientific studies. I think maybe you've heard a lot of news that scientists are trying to replicate a study, they're trying to reproduce a study, yet they're not able to. They're failing to do so. And of course, as we said, reproducibility is important for reliability. So if you have a crisis, this means that there will be concerns about the reliability and integrity of published research findings. The crisis actually gained widespread attention following several landmark events. The first one was in 2011, where Bayer, which is a pharmaceutical company, tried to replicate 67 published studies on oncology, women's health, and cardiovascular indications. They found that only 25% out of these 67 published studies that were published in reputable journals by reputable scientists, they found that only 25 of these published studies could be reproduced. Then in 2012, the next year, Another pharmaceutical company called Emigen found that only 11 out of 53 published studies on hematology and oncology were reproducible. Again, these were studies that were published in reputable journals by reputable scientists. Then in 2015, the Center of Open Science did a reproducibility project, especially targeting psychology studies, and it found that only less than 50% out of 100 published psychology studies were reproducible. And then in 2016, Nature did a survey where it surveyed more than 1,500 researchers. And the survey found that 70% of them failed to reproduce another scientist experiment. 70% of these researchers tried to reproduce an experiment or a published study, and they failed to do it. And more than 50% of them failed to reproduce even their own experiments. So even work that they did themselves, they were finding it difficult to reproduce. So as you can see, that this raised a lot of alarm bells. The scientific community was not really paying much attention to reproducibility, but these events really sounded the alarm bells to the issue. And in this Nature survey, it actually found that despite the evidence of the lack in reproducibility, 73% of the surveyed researchers think that at least 50% of the papers in their field can be trusted, noting that they've just said that 70% of them said that they couldn't reproduce other people's work, and more than 50% of them said they couldn't reproduce their own work. But when they were asked, like, so can you trust, that the scientific literature in your discipline? The majority of them said that yes, at least 50% of this can be trusted. And this is, of course, is alarming, because it means that there are greater efforts needed to enhance the awareness of researchers on the reproducibility crisis, and on the danger of blind trust on published research literature, because the traditional publishing system have made us think that if a study is published in a reputable journal, then you can 100% trust it because it has been peer reviewed. This journal will probably not publish a study that is not good. But the fact that the truth is that a lot of published studies are really not reproducible. This is really big issue, and this should be an eye opener that you shouldn't just trust a study because it's published in a journal. You should have apply your critical thinking to evaluate this study and make sure that you can really trust the study to build on it. And I really love this comic, and it mainly depicts that mistakes in peer-reviewed papers are easy to find, but they're hard to fix. So as you can see here, the scientific publishing, the traditional scientific publishing is like a fortress, and the researchers are trying to sound the alarm that there is an issue, yet the fortress is just, the doors are closed. A study is published, then it's really difficult for it to be retracted. If you go back to a session that I did earlier here in the club, was about the issues with the traditional publishing system, is that there are a lot of retractions. Each year, more and more papers are being retracted from journals because they are not, they have major issues in them despite them being peer-reviewed. But this retraction takes years, and while these studies are still published, scientists are using them, they are building on them, while the work is actually not really robust. So then the implications of the reproducibility crisis would be simply that the public will lose trust, and we've seen that during COVID. The public started to lose trust in scientific research. There's a lot of waste of resources because scientists are building their work on research that has flaws in it and results that are not particularly true. And this will, of course, lead to scientific stagnation. People will just be circling the same circles, and scientific growth will be halted. So you might wonder what are the causes of the reproducibility crisis, and there are so many causes, but we'll try to talk about the most important of them. So the first one is the poor methodology. That's basically it. Very poor methodology. We have statistical errors. We have publication bias. We have pressure to publish. We have lack of transparency. And all of these kind of work together to cause the reproducibility crisis that we're currently seeing. So we'll take each one and we'll try to discuss it in more detail. So poor methodology, we see that scientists are in a rush to do the studies, so they don't really take the time to do good experimental design. Many studies don't have standardized protocols, even though that many experiments don't have standardized protocols, even though that there are experiments that are used daily by thousands of researchers, yet there is no standardized protocols for these experiments. And when the methodology is published, there is no sufficient details described in the methodology part. Because we're usually limited by a word count, or maybe we don't want to share the details of our methodology, so some details are lost. And this will lead to confounded and biased results, this factor of poor methodology. Then we have the statistical errors, and we have misapplication of statistical methods. And I always remember Professor Fahal always saying that you should always have a statistician as a friend, because only the statistician will be able to tell you the proper statistical test that you need to do. And this should be done before conducting the study, because you need to know what data you need to collect, what you need to do so that you can reach or you can do good statistical, use good statistical methods. And unfortunately, also some scientists are manipulating statistics to find significant results. So, for example, we have things like the p-hacking. We also, many studies have small sample size due to many factors, but this leads to low statistical power. So what this means is that we will also come up with incorrect conclusions that will lead to the results not being reproducible. The third thing is the publication bias. What we see is that the traditional publishing system prefers publishing positive and novel findings, while they neglect negative or null results, even though that these negative results are as important as the positive results, because it shows that this work has been done and it didn't work. So why would another group of researcher or another researcher waste time and money doing the same experiment only if it was already published that, some scientists would say that we did this experiment and we got negative results. But the publishing system prefers positive results and prefers novel findings. And this, of course, skews the understanding of reality, making scientific literature based towards success. If you have a negative result, it means that you are not a successful scientist, you're not a good scientist, while this is not the truth. If you get a negative result, it's a result at the end. Then we have the pressure to publish. The traditional publishing system or the traditional publishing atmosphere really say that you should publish or perish. And what this does is that it puts pressure on scientists to rush studies to cut corners, and to manipulate the results. And again, unfortunately, this will result in results that are more appealing to high-impact journals, but they could be non-reproducible results. Finally, we have the lack of transparency. Unfortunately, there is a big issue in access to raw data and code, if code was used in analysis. And many scientists don't disclose all the relevant experimental details in their published studies. So what this does, it also prevents other researchers from accurately replicating your studies. If I don't know, if I cannot see your data, if I cannot see the code that you use for the analysis, if I cannot see the details that you use in the methodology, then I will not be able to replicate your study. So the question is, how can research be more reproducible? And the answer is simply, we need to move away from these factors that lead to research not being reproducible. So, and we can see that utilizing good and open research practices could help us move away from these issues that cause the reproducibility crisis. So, for example, if we have poor methodology, one of the solutions is standardized protocols. You know, if there's an experiment that scientists are using all over the world, then it might be a good idea to have a standardized protocol that everyone in the world would adhere by. Then it's important to have comprehensive documentation of methodologies and procedures. You know, you shouldn't just write a few things about the methodology you use. You should write every nitty-gritty detail of what you use during your study. And this ties into open reporting of methodologies and procedures. And open science is trying to move people towards open methodologies. There are many platforms such as protocols.io that allow scientists to publish the details of their methodologies. Now there are even open lab notebooks. So even the notebook that we use in the lab that we scribble on while we're doing the experiment, even those you can make them open for everyone to see, so that they could see the details even of the day-to-day experiments that you did. Then we have the statistical errors. Education and training is very important. You know, there is a lack of education and training when it comes to statistical techniques. So it's really important that before we conduct the study, we really look into the statistical analysis of similar studies, talk to a statistician, and make sure that we're taking everything into an account, so that at the end we don't really have to, you know, cut corners to achieve a significant p-value. It's also important to advocate for standardization of statistical reporting. Currently, there are, like, anyone could report on their statistical analysis the way they can, but if we have a standardized way of reporting, then this might make things more clear and more reproducible. And of course, the most important thing is robustness. You need to use robust statistical analysis. Then we have the publication bias. What we should start doing is report all results, including the negative or not finding, because they are as important as the positive results. We need to promote the use of pre-registration and registered reports. And these are also practices that Open Science is trying to encourage people to do, is that before conducting the study, you publicly make your hypothesis available, you publicly made your data collection protocol available, you publicly make your methodology available, and this will prevent us from, you know, trying, if things didn't work, we will have to report that this methodology did not work because we've publicly said that we want to use this methodology. But if it's not pre-registered, or the report, we don't have pre-registered reports, then you know, we can just simply change the methodology. We don't have to report on the methodology that didn't work. And the pressure to publish it's really important that we start promoting quality over quantity and impact of research. We really need to start moving away from journal impact factor and encourage the use of alternative metrics or altmetrics. This is also part of what Open Science is advocating. For example, we don't see what's the impact factor of the journal that the article is published in, which doesn't have to do anything with the article, but we see the downloads of the article, we see the views of the article, we look at whether the articles have been, for example, used in policies, whether they have been mentioned in media, even in social media. So this is what Open Science is trying to move towards. And it's also important to promote collaboration over competition in research, because together people can do much more robust work, and they can do better work than instead of competing on resources that might already be limited. Then finally, we have the lack of transparency, and Open Science is basically trying to completely abolish that. Open Science is trying to make all of the research process, methodology, the data, the code, everything to be shared publicly and openly for everyone to see. Also encourage open peer review and critique, because currently in the traditional publishing system, the peer review is like a black box. People reading the article don't know what the reviewers said, what the changes that were made to the manuscript, who were the reviewers, maybe there's a conflict of interest. So it's really important to make the peer review process open. And also, Open Science is trying to encourage the use of open scientific infrastructure such as OSF and Octopus, and these are basically platforms that would enable you to really make all of your research process open and shareable to everyone who wants to use them. So again, I love this illustration by the Tuning Way. The road to reproducibility is not straightforward. It needs that you make everything open, from your data, from your code, from the tools you used, and from your results, even if they were not significant, even if they were not positive. And this is almost the only way that reproducibility could be achieved or ensured. So talk about some of the effort that some of the stakeholders are doing to promote reproducibility. So we have funding bodies. Many funding bodies are currently requiring grant application to submit a data management plan, to submit a pre-registration, to submit a registered report before this project could be funded. Again, as I said, they require pre-registration or registered reports, and even some grants are even currently, some funding bodies are giving grants for replication studies. So now this is becoming a type of study that funders are attracted to, because it is really a crisis. We also have publishers and journals. Many journals now require you to adhere to some reporting standards. For example, for clinical trials, we have the CONSORT guidelines. They require you to make the data open and publicly available as a condition for publication. And some of the publishers are currently offering open peer review process so that it increases the transparency and the accountability of the peer review process. So I guess the key takeaways is that reproducibility and replicability are essential for validating scientific findings and ensuring research integrity. There are key factors that are contributing to the reproducibility crisis, mainly the lack of rigor, inadequate peer review, and restriction of access to data. And open science practices like sharing data, preprints, open peer review can really address the core issues or core factors that are causing the reproducibility crisis. And at the end, this is a small quote, is that in the world of science, the reproducibility of research is what makes it reliable. So let's commit to making our research not just innovative, but also replicable and transparent for the generations to come. And with that, I come to the end of my presentation, and I hope I didn't take too long. And I'll be happy to receive any questions you have. Thank you for listening.
Thank you so much, Lemise, for this. This was very informative, and I love the quote by the end that you concluded your presentation to. Thank you so much. So now the floor is open for questions. If you have any question, you can raise a hand, and there are already some questions in the chat. So do you think we shall start by the questions in the chat until we have people raising their hands?
Yeah, sure. Yeah.
Okay. So one of the questions was asked by Dr. Abdullah. He asked about examples for reproducibility and replicability.
Okay. So as you said, for reproducibility, you're going, for example, you see a published study. They use, for example, I see Dr. Abdullah have his hand raised, so maybe he wants to add to the
Dr.
question.
Abdullah, you can go ahead.
Thank you very much, Lemise, and thank you, Samira, for this opportunity. Actually, I'm asking this question to make it clear that sometimes, even if you are using the same methodology, the detailed methodology, but you are using a different data set or a different patient materials, you might get a different result. How can we call this is not a replicable data? While we know that, for example, a set of organisms or a set of gene data can be reanalyzed using the same methodology, the same setting of the bioinformatic software, our tool, as example, using the same computational environment, but again, you get a different result because you are handling different set of data. So reproducibility, I would like you to give you an example, but for replicability, I gave you already the example. I need your comment on that. Thank you.
Thank you, Dr. Abdullah. Yes, and replicability, it's because you're using a different data set. For example, let's say you used some genes of Europeans, and you want to do the same study on genes of Africans. Then there is a big probability that you're going to find different results. So this is okay because you use different data, but you use the same methodology, and this is okay because this is justifiable. You change the population, and this means that this result cannot be generalized on the entire universe. For example, you can confidently say that this, for example, this intervention works for Europeans, but it doesn't work for Africans. Because the results are not replicable when it comes to the African community. So this is replicability. It's okay that the results are not the same at this case. But in reproducibility, you're using the same data, you're using the same methodology, but so in this case, if you get different results, then there are question marks. What happened? Did the authors of the original research not really, they missed some details that really had a change on the results? Maybe they did some, p-hacking, for example, and this is evident, and this is the reproducibility crisis. So all of these big pharmaceutical companies were trying to reproduce published results using the same data, using the same methods, and they were not coming to the same finding or same conclusion. So I hope that this clears it up, Dr. Abdullah. Is Dr. Abdullah not online?
Not sure. I think he has some network issues.
Okay. I hope I answered the question.
Okay, we can go to the next question. We asked is that how can we ensure reproducibility of research during the urgent epidemic situations, for example, the COVID-19? where decisions like vaccine development depend on rapidly generated but high robust data?
Yeah, it's by taking the measures that we discussed, making everything open. Every nitty-gritty detail of the data you used, every nitty-gritty detail of the methodology you used, the statistical analysis you used, everything. If everything is open, then let's say I want to see if I can trust your research, I can easily try to rerun your analysis, or even if I couldn't do it, someone else would do it. And if it's not reproducible, they would report like, this finding is not reproducible. And we saw that a lot in during COVID. Many people were publishing, and I think this also we discussed that in the issues with the traditional publishing system, because during COVID, magically the publishing delays, when you submit from the time you submit your paper to the time it's published, it usually took years, but like magically during COVID, it took weeks. The publishers were suddenly able to just publish these results. So this led to a lot of not very good research being published. And many scientists were able to catch that, were saying that this result is not reproducible. So I think for me, it's just openness. Making everything open would make people being able to trust your research more. I hope that answered the question. I can see Dr. Abdullah is back.
Yeah, Dr. Abdullah is back. Yes,
Yeah,
Dr.
sorry, I missed actually your answer because I lost my connection. But
No
regardless of your answer, I would like to comment also on the latest point you just raised. I don't agree with you that every data will be replicated, because we have two things here. We have the reproducibility of the method itself, because many methods known to have a very poor resolution and poor reproducibility. I remember maybe 20 years back when we start using PCR-based genotyping of bacteria. This genotyping actually were based on very small alleles. So it's very easy in bacteria to lose this allele, or you get a new SNPs, which make two different isolates looks like identical and clonal. But when the resolution of the method has been improved by introducing the DNA sequencing, we used to get a better result. But even though during the crisis of the corona, we had a very high pressure on the virus to develop mutation in the spike protein, and because of the many cases which has been occurred during a very short time, and this is why the mutation rate and the detection of this mutation was very high. So again, I can use the same set of data, same methodology, everything, and again, I might not get the same result because of a very small sequencing error. So something related to the method itself. So first of all, we should agree that the method we use in research, every method, if it's a laboratory method, or for example, collecting data, clinical data by interview, or assessing patient clinical assessment, whatever the type of tool we are using for conducting research, every single method have a limitation in reproducibility. So even if you replicate the same exact methodology, you might not get the same result in the same center. But still, for example, if we have a multi-centers clinical trials, if we evaluate a new treatment for treating, for example, a certain infection, for example, we might get 90% success rate in one center using the same methodology, and maybe 88% in other center. And again, this is a very good result, a very good reproducible result to get very close. We cannot get always 90% efficiency in treating the patient because there is so many other factors which can affect our results, even if it's only the exact methodology. So we think about the different setting. If you do the same test, use the same research tool in the same center, you might get different results. How about doing the same in different centers and different settings and using the same methodology? I agree with you in the point of the open science that we need to share everything. But even though if we share every details about the method, nobody can guarantee that the same result will always appear. This is the first thing regarding the reproducibility. If you are using the same set of data, analyzing the same set of data in different settings. But when it comes to different set of data, definitely nobody will ensure that you are going to get the same result. And I will draw your attention to one major thing in research right now, which is a personalized medicine. One drug cannot work for every other drug. So if I get a good result using some clinical trial setting, and I say that this is works for the European people, for example, nobody can guarantee that it will work for another European people from the same setting. Think about another diverse group from Africa or from Asia. I don't think we should focus a lot in replication of the same result, but we should say we need to be an open in science, sharing all the details, hoping that we are going to get closer results. But nobody can speak about replication of the same result and replication or reproducing the same details by the same technique. And we are in chemistry, for example, clinical chemistry. If we are measuring blood glucose, for example, every single method have a variation which is acceptable. So if you take the same sample and you test it 20 times, you are not going to get the same result. You are going to get plus or minus variation from the mean. And we accept that. We accept that, for example, the blood glucose estimation by this method have 2% coefficient of variation. So we cannot, and this is done by the same equipment, the same technician, the same lab setting, everything the same. And we still are getting differences in the results. But I think the good takeaway message from your lecture is we need to be an open in science. That's all. If you are open in science, we will be very close to each other. Thank you very much.
Thank you, Dr. Abdullah. I agree. We cannot say that the result would be 100% reproducible. For example, let's say you have a patient and you're taking their blood glucose, you measured it, you took a sample, you measured it, and it was normal. You measured it again, from the same blood spot, and it was not normal, it was high, and then you measured it, it was low. This should give you an indication that there is an issue. Maybe there's an issue with the equipment, maybe there's an issue.
But there is a limitation, but there is an acceptable limit of variation.
Exactly.
We cannot we cannot accept, for example, for a qualitative variable to be sometimes positive and sometimes negative. But for quantitative, we have a margin of plus or minus. And this plus or minus usually published with any kit and any diagnostic tool. For example, if you are measuring viral load, the company will tell you after validation of the kit that the viral load limit, it detects, for example, 500 copies. If the viral is less than 500, it won't be detected at all. So this is the idea that every single research tool have a limitation, and we should accept that. Otherwise, we are not going to conduct any research.
And this is not what we're targeting when we say we have a reproducibility crisis. When we say that the reproducibility crisis is that we use the same data, we use the same methodology, and we get completely opposite results. Let's say we do an analysis on a certain, on the same set of genes, and we find that we have a SNP that's, common among this community. Then another scientist will come and use the same genes and do the same analysis and doesn't find the SNP is common among this community. And this is what was happening.
We do have two sides of error sources. We have something related to the method and something related to the samples itself. Yes. Okay, if we standardize everything in the methodology side, we're still lacking the standardized, or we cannot standardize actually the sample, even if it's the same sample. But it's a good idea that we need to be as reproducible as possible.
Sample, like for the sample, we're using the same sequence sample. You did the research, you sequenced some genes in a community, and did the analysis and got this result. And I came and I used the same sequence.
I understand you. I got you. Yeah, I got. I remember. Lamis, I remember Lamis. Maybe 20 years back, I was working with a very intelligent bioinformaticians He's from the Netherlands, and he told me, but using statistic, you can prove anything and you can reject anything.
Yeah.
Just by playing in the biostatistic tools.
Exactly.
No?
Exactly.
So this is the idea. We need to fix the analytical side, the tools of the research we are using. Like if we are using a bioinformatic tool, yesterday I did a bioinformatic analysis and there was two options. Option 1 and 2. First, I started with option 1. It gave me different result. And I said, no, I should use that option 2, and it gave you completely different result. So the setting of your research tool, where you analyze your data or you collect your clinical information, it needs to be standardized. But again, the other part,
stated openly.
Yeah. You should make it clear, as you said, and you should disclose everything. But still, we have other confounding factors which is going to affect the outcome of your result.
Definitely.
Thank you, Dr. Lemis. Thank you very much.
Thank you, Dr. Abdullah. Thank you.
Well, I'm really enjoying it's
Samira, your voice is breaking up. Or is it only me? Samira is out, but I see Emmanuel. Emmanuel Bache is the executive director of the Africa Reproducibility Network. And Emmanuel, I think do you want to comment on something?
Yeah, I think I wanted to comment on what Dr. Abdullah said. I wanted him to know that this is something that the research community is quite aware of. I'm going to share a paper in the chat that kind of further explains what he just said. I think in every research, there are the known unknowns and then unknown unknowns, right? They are called hidden moderators. And that is what Dr. Abdullah was talking about them. And I think these become more prominent when you are trying to replicate a study. I think reproducibility is quite simple. You just take the data that they presented and then the same code. So what you're essentially doing is that you are just rerunning the analysis. You are not doing anything new. And that is where during that, you are expected to maybe get the same results. But even that, there are various arguments that are raised against it. I think for anyone that enjoyed the conversation right now that Dr. Abdullah and Lamesh had, I'm sure you're also going to enjoy the paper that has been shared. I think it presents a lot of reasons why reproducibility and replicability are not perfect and conclusive, and the fact that there are a lot of reasons why you need to look at various factors when you're trying to do any of them. Yeah.
Thank you, Emmanuel.
Thank you, Emmanuel, for sharing this.
Samira, we lost you for a few minutes.
Oh, okay. So I was just telling you that it's really nice to hear other opinions and other perspectives. I really enjoyed the discussion that you had with Dr. Abdullah. And thank you, Emmanuel, for sharing the paper. There was a question in the chat from Amna, I think. So she was asking about in reproducibility, is the mistake from the analysis process? Like if you use the same data and the same methodology?
It could be in anything. It could be in the methodology. It could be in the analysis. So there is a question mark, it's if the results are not reproducible, you used exactly the same data, you took into account try to control all the possible confounding factor, and you run the same analysis, then yes, probably there's an issue somewhere in the analysis. Maybe the authors, as Dr. Abdullah just said, like for example, in things like bioinformatics, like if you change one parameter in one type of analysis, it could give you different results. So maybe the scientists did not clearly state which option they chose. So there is an issue. There is an issue with the analysis. Yes.
Okay, that's great. So if anyone has a question, you can feel free to raise a hand or write it down in the chat. Lemis, in the meantime, regarding the open lab notebooks, are there different lab notebooks depending on the different specializations? For example, like people working in physics don't use the same lab notebooks as those in chemistry and so on.
Yes. Different disciplines have different lab notebooks, but now there are some platforms that just allow you to scan even the lab notebook that you scribble on, write on, during your work, you can just scan it and make it open for people to flip through. But this is definitely because more people, for example, some people use code, some people don't. So yeah, different disciplines have different notebooks. Yeah.
Okay, it's really nice to hear this. And Sadiq has a question in the chat. So he's asking that we're seeing an increase in publications from conflict zones. So some of this work is new and interesting, but it also raises concerns about the research quality and integrity during this period. So what is your view on this issue, and what do you think will happen to these publications?
I think it would be unfair to judge a publication quality based on where the publication is coming from. This is a big issue, and it's been a huge injustice for us in Africa, for example. Like many journals, you submit a paper and they see that all the researchers are from Africa and they're not known, then this is judged as low quality, instantly. This is not fair. Again, you should be the critic. As we said, you should not blindly trust that a paper is published, is peer-reviewed. This means that it's of good quality. You should be the critic. You should go and really go into the methodology, go into the data. If there's a possibility for you to rerun the analysis, for example, if it's just like a code that you can execute and you have the data, then do it. Like if it will not take a lot of resources from it. So it basically comes to you and your judgment. And of course, the more information you have when it comes to that research, if the data is open, for example, if they use a survey, the survey is there, all of these factors, if you have them, then this will make you more confident that this research is good. But I think it's really unfair to think that because it's coming from a conflict zones and then its quality is poor.
Okay, this is actually this is a big question because people from low-income countries, not only conflict zone areas, like there is some bias towards the researchers coming from these regions actually.
Yes.
Yeah. So if there's an additional question, we're actually 4 minutes beyond our time. So Lamis, I don't know if you have some time to stay.
Yeah, it's okay. If there are other questions, then I'm happy to stay.
Okay, so if there is any question, you can feel free to raise a hand or write it down in the chat. Well, I can't see any raised hand. So would you like to add anything, Lamis?
Yeah, I would just like to say that we're not saying that open science is the ultimate solution for all the issues that are currently happening with the publishing system. Again, and this is what we encourage our community to do, is that not take everything for granted and say that this is the gold standard, because as Dr. Abdullah said, each situation have its own measures that you need to take. So we're just saying that open science is one of the tools, of the many tools that could help you, make research more reproducible. But it's definitely not the only solution or the best solution. I'm sure as times go, more and more issues are going to arise and more solutions are going to arise. I don't know if Emmanuel wants to say something.
So, Lamis, we really enjoyed the discussion today. I will reshare the. Oh, I'm sorry, I didn't hear you well.
No worries. I was just throwing Emmanuel under the bus and asking if he has any final remarks to say.
Oh, okay, Emmanuel. Emmanuel, it was really nice having you today.
Yeah, it was great. I mean, Lamis shared the link, so I tried and joined. Not much. I think Lamis covered everything. And what we would say is that some of these things can seem a bit new and overwhelming. We already know that researchers have a lot on their plate, and sometimes trying to apply all of these principles can seem like a bit too much. But I think what we always say to every community is that you take what works for you, and you don't need to do everything at once. Just start small, and then keep on applying them bit by bit. I think the goal of open science and all these principles is to make research more efficient, more effective, and more impactful. So it's good for all of us, but adapt it to your need and just stick to the core principles here.
Okay. Thank you, Emmanuel, for this. Thank you so much. So, Dr. Abdullah, if you'd like to add anything before we conclude.
No, thank you very much. Thank you very much for everyone.
Thank you, Dr. Abdullah. So by getting to this point, we would like to conclude our session. It was really nice having you all here today, and big thanks to Lamis, our presenter for today. And we look forward to seeing you in the upcoming Mycetoma Research Club sessions. And until then, stay safe and have a wonderful time. Bye-bye.
Thank you. Thank you, Samira. Thank you for having me. Bye.
Bye.
Have a good day, everyone. Bye-bye.